so much for the kind introduction.
So my name is Vera. I'm a PhD candidate at the Cambridge Centre for Medical Materials and an honorary clinical fellow at Cambridge University Hospitals. And my PhD focuses on the artificial creation of human heart tissue. So as a clinician, this is what I'm interested in, and this is what my funding body is interested in, the British Heart Foundation. Throughout England and Wales, every day, 530 people will go to hospital with a heart attack, and 190 people will die as a consequence of this heart attack. So once you have a heart attack, this is what your ECG looks like. I'm sure most of you will have seen something like this before. It's this very significant peak over here. And this peak over here is caused by your heart looking like this. So we're all familiar with how a heart attack happens. You have a plaque starting to build up within your artery. It gets bigger and bigger over time. At some point, there is a rupture, a blood clot forms. And then over here, this is an actual human heart. You see the scar formation. So this is a relatively fresh heart attack with the bleeding on this side here. So the problems this causes are, manifold. You will end up with a lower ejection amount of blood from your heart. At the moment, this can be treated, not fixed, with so there are two big ways of fixing this. One of them is the actual intervention. Where does it go? There. The actual intervention. So we put in a stent, we put in a bypass graft, we make sure your heart muscle gets blood again. If this is not an option, the other way of doing this is by medication. So we prescribe aspirin, beta blockers to kind of help your heart, along. Both of these treatments are not ideal because you will end up with scar formation in your heart and with a limited heart function. So what I'm working on is this third part down here. So I specialize in something that is called regenerative medicine, which basically builds a plaster of human heart tissue to be directly applied to your heart after the heart attack to revive the tissue underneath and integrate. So to be able to do this, at the beginning of my PhD, I needed to think about how, so in which environment human heart cells live. And in order to do this, the best way is you go to a butcher and you buy yourself some pig hearts. They come at £3 per piece. And then you get like these lovely little cylinders down here you see on the right. So in this cylinder, you have the extracellular matrix, the home of the heart cells, and you have the heart cells themselves. So to get rid of the heart cells and just be left with the home of the heart cells, I put these hearts through a chemical washing machine, and I end up with something which is basically just collagen and sits down here, this perfectly white, lovely little marshmallow. If you stain this, so just to explain these images down here, if you stain this with something that detects DNA, so what is in the nucleus of a cell, it goes blue. So you can see here, this is this sample actually. You can see that there is nothing blue here, and if I overexpose it, I get a bit of background staining, so this is proof that the staining worked, but there are no cells in there. And if you actually quantify the DNA within this sample, throughout its decellularization or washing process, you can see how the amount of DNA goes down. This is relevant for immune reactions and all these things. So lovely. We're just left with the home of the heart cells. So next up, I took this and I put it into a CT, like we have down at Addenbrooke's, the big machines they put you through if you break something. So I put it through the CT, and this is what it looks like. So it's a lovely hollow structure we were able to wash out. And because now I'm more of a material scientist than a clinician, I ran these samples through the whole battery of material science analysis. So if you take this lovely thing over here and you put it into water, because obviously it will be in an aqueous environment after we implant it, You can see how it takes up water over time. And if you focus on the left here for a second, you will see blood vessels coming through. They're the big one. So, which is great, because we were able to preserve the internal structure of it, and we're able to study it. If we take this and we put cells on it, if we plot this here against our positive control, so red is what these cells normally live on and are most happy on. So this, obviously the natural environment here, they will enjoy more than they do the artificial tissue we grow them on. So this is what it looks like as a cross-section. Here again, the vasculature, the preserved vasculature. And this is what it looks like under a scanning electron microscope. And for various reasons, we cannot use these to actually implant into humans. So we have to come up with a safe medical standard version of them. And what I came up with over the course of my PhD is this, which I'm very proud of and was very difficult to get. And if I put it through the same battery of things, this is how I can modify it. So you see in red is the natural decellularized version of the marshmallow I showed you earlier. And these are all different versions of the structure I made that we can produce. So we are, at this stage, very happy with the home of the cells that we have identified and recreated. So what we do is we populate them. Does it do it? Yeah. And we populate them with this. And those are embryonic stem cell-derived heart cells. And if they are really happy, they start to do this. About 7 days down in differentiation, they start to beat spontaneously in culture. And so the big question of my PhD was, if I get enough of these cells, and if I put these cells onto my artificial scaffold, will they be happy? And I'm happy to say they are. They actually started to beat coordinatedly in 3 dimensions, and we're going into in vivo experiments within the next year. Thank you.
Vera, thank you. Absolutely stunning. We've got time for 1 or 2 questions, if you would like. No, you've completely satisfied. Oh, yes, please.
So the structure I made is from collagen. It's pure collagen. So you can buy this from Sigma Aldrich or Collagen Solutions, and it comes in a powder. So what we do is we process it with a technique called crystal templating. So we basically freeze a collagen suspension, and the ice crystal, we can control the growth and the direction of the ice crystals that form during this freezing process. And then we sublime it, so we use the crystal as a negative to create the porous structure. If you can imagine, this would be where the crystals sit. And after we get rid of them, we're left with these really delicate collagen walls.
Wonderful. I think we're all stunned, Vera. That's wonderful. Thank you very much indeed.